Okay, let's unpack this. Imagine a future where our beloved dogs and cats, they're not just our companions, right? But they're also patients. Patients on the very front lines of genetic medicine. We're talking about something really quite profound here. Gene editing in companion animals. Now, this isn't about, a new dietary supplement or even some cutting-edge vaccine. No, it's about fundamentally changing the DNA blueprint of our pets. Like, quite literally, erasing diseases right at their source. Now, if that concept sounds bold, maybe even a little bit like science fiction, well, it's because it absolutely is. I mean, this frontier, this area of biological intervention, it raises incredibly profound medical, scientific, and ethical questions that we as a society are really only just beginning to grapple with. So our mission for this deep dive is to, well, thoroughly navigate the intricate science behind it all. We want to explore its incredible potential, to maybe alleviate suffering, but also confront the significant technical and biological challenges that definitely still exist.
And finally, try to unravel the complex ethical landscape that this kind of transformative power introduces. For this deep dive, we've been immersing ourselves in some really insightful excerpts from discussions and research, things titled CRISPR for Companion Animals, Ethics in Future, and also Pet Gene Editing, Reshaping Animal Health.
So let's begin our exploration into a future that's, well, maybe closer than you think.
Right. To really get a handle on the revolution we're discussing, we absolutely need to start with the basics. The foundational technology, the one that sort of kicked all this off, is CRISPR-Cas9. And even with all the exciting new advancements we'll definitely touch on, CRISPR-Cas9, it really remains the workhorse of gene editing. It's fundamental. It's a technology that truly revolutionized molecular biology when it, burst onto the scene back in 2012. It just made gene editing so much more accessible and efficient in a way that was, well, previously unimaginable. Before CRISPR came along, gene editing was much more difficult, really cumbersome, expensive, and frankly less precise. People relied on older technologies like zinc finger nucleases, ZFNs, or TALENs, and those were just difficult and really time-consuming to design for specific genetic targets. CRISPR, well, it changed the game entirely. So to simplify how it works, think of it as a highly precise biological scissors system, operating right down at the molecular level. Imagine you want to find and maybe change a very specific sentence in an incredibly long, complicated book. Let's say the entire genome, which for a dog can be billions, literally billions of DNA letters long. Within this CRISPR system, there's a really crucial component. It's called a guide RNA, or gRNA for short. This guide RNA, it acts like an incredibly sophisticated GPS. It really does. It's a short sequence, and it's designed to be perfectly complementary, like a matching key to a very specific DNA sequence you want to target. So it meticulously leads the Cas9 nuclease, that's the molecular scissors part of the system, the enzyme that actually does the cutting. It leads it directly to that precise DNA location within this vast genome.
Okay, so the guide RNA finds the spot.
Exactly, finds the exact spot. And once the guide RNA has positioned the Cas9 enzyme correctly, Cas9 does its job. It creates what's called a double-strand break in the DNA.
A double-strand break.
Yes, it literally snips both strands of the DNA molecule, right at that exact predetermined spot. Now, once that precise cut, that break, happens, the cell doesn't just sit there. The cell's own natural repair mechanisms immediately spring into action. It wants to fix that break. Evolution has equipped our cells with these incredible ways to fix damaged DNA, and gene editing, well, it cleverly exploits these existing pathways. There are two main ways the cell tries to repair this double-strand break, and both can actually be harnessed for therapeutic purposes. The first is called non-homologous end joining, NHEJ.
NHEJ.
That's right. And you can think of this as a bit like a quick and dirty repair job, a bit rough and ready. The cell essentially tries to glue the broken ends back together as fast as possible, but it often does so imperfectly. This frequently leads to small random insertions or deletions right at the break site. These are known as indels.
Indels, insertions or deletions.
Exactly. And while this sounds imprecise, maybe even messy, it's actually incredibly useful in many cases. This process typically results in the gene being disabled or, as scientists say, knocked out. So it effectively turns off a faulty gene that might be causing a disease. It stops it from producing its protein or just disrupts its function entirely.
So breaking it is sometimes the goal.
Precisely. If a disease is caused by a gene that's, say, overactive or producing a harmful protein, simply breaking it, disrupting it with NAJ, can be a really powerful way to turn off that problem gene. Then there's the second repair mechanism. This one is often more sought after, especially for specific corrections. It's called homology-directed repair, or HDR.
HDR. Okay, that sounds more directed.
It is. It's a far more precise and, you could say, elegant repair pathway. If, along with the CRISPR machinery, you also provide a template DNA sequence to the cell, a template that contains the desired genetic information you want to insert or correct. The cell can actually use this template as a precise guide during its repair process.
So you give it the instruction manual for the fix.
Exactly. Think of it like meticulously rewriting a specific sentence in that giant book, ensuring the new text is perfectly aligned with the correction or the insertion you want. This HDR pathway, it allows for the precise correction of existing genetic information. You can fix a specific point mutation, a single typo, or you can even insert entirely new genetic material into the genome right at that predetermined location. This pathway is really where the true promise of gene correction rather than just gene disruption lies. It lets us mend specific errors.
That's a fantastic breakdown of the foundational CRISPR-Cas9 system. Really clear. But, here's where it gets really interesting and where the field has just evolved at such an astonishing pace. For a while, CRISPR-Cas9 was, undoubtedly the star. And it still is for many things. But researchers, they quickly realized its limitations too, particularly that risk of indels you mentioned, or even those off-target cuts, hitting the wrong spot. So the scientific community, always pushing forward, driven by this need for even greater precision and control, they've moved beyond just Cas9. They've developed even more refined tools that offer remarkable specificity and flexibility. We're talking about advancements like base editing and prime editing. These really represent a new generation of gene editing, and they promise even more tailored approaches to genetic diseases.
That's absolutely right. These newer tools, they truly exemplify the rapid innovation happening. It's incredible. Take base editors, for instance. They offer a much more refined approach than just cutting DNA. Their mechanism is, quite ingenious, actually, and it bypasses the need for that double-strand break entirely, which is a big deal.
No double-strand break. How does that work?
Well, these systems, they contain a deactivated Cas enzyme, so it's been engineered so it still binds to the target DNA, but it doesn't actually cut both strands. They combine that with something called a deaminase enzyme. So instead of creating that break and then relying on the cell's sometimes messy repair mechanisms like NHEJ, they chemically convert one nucleotide, one of those DNA building blocks, directly into another. Specifically, they can change a cytosine, a C, to a thymine T, or an adenine, A, to a guanine, G.
So it's like chemically rewriting a single letter.
Exactly. Think of it not as cutting and pasting, but more like using a very precise chemical erasure and pen. Changing just a single letter in the DNA sentence without tearing the page, without causing any larger disruption. And the key advantage here is, it's profound, really, because this process avoids creating those double-strand breaks that traditional CRISPR-Cas9 makes. It means significantly fewer random insertions or deletions. Fewer of those indels occur. You get a much cleaner, much more targeted edit with potentially less collateral damage to the surrounding DNA.
Okay, so fewer mistakes, basically.
Fewer unintended mistakes, yes. This reduced off-target activity and increased precision are huge steps forward for therapeutic applications, especially when you're trying to fix a single specific error, which, is often the case for many inherited diseases. It's about achieving surgical precision, but right down at the molecular level.
Wow, okay. Now imagine something even more sophisticated then, like a find and replace function for DNA, but one that can handle not just single letters, but maybe entire words or even short sentences. That's essentially what prime editing brings to the table, right? Taking precision, another crucial step further. It really feels like a quantum leap in genomic control.
It really is an incredible advancement. Prime editing is extraordinarily sophisticated. It employs a reverse transcriptase. That's an enzyme known for its ability to synthesize DNA from an RNA template, almost like a molecular scribe, and it fuses that to a Cas9 nickase.
A nickase, not the nucleose.
Right, a nickase. Crucially, unlike the full Cas9 that makes a double-strand break, a nickase only cuts one strand of the DNA. It just creates a nick rather than a full break.
Ah, okay. Less disruptive.
Much less disruptive. And this system also uses a specialized guide RNA called a prime editing guide RNA, or pegRNA. This pegRNA is clever. It contains both the guide sequence to locate the target and the template for the new DNA sequence that needs to be inserted. So this combination allows for a true search and replace capability within the DNA. It's quite amazing. The pegRNA guides the nickase to the target site. The nickase cuts just one strand, and then the reverse transcriptase uses that pegRNA as a template to directly synthesize and insert new DNA into that nick strand. It seamlessly replaces or adds specific genetic information.
Wow, okay, that sounds incredibly precise.
It is. It's like carefully unzipping just one side of the DNA ladder, and then a tiny molecular scribe, the reverse transcriptase, comes in. It uses a specific template, the PEG RNA, to write in new information, seamlessly replacing the old. Now, for veterinary applications, this remarkable specificity is particularly important because many companion animal diseases are caused by single base mutations, or sometimes small insertion-deletion mutations, things that might be too complex for base editing but maybe too small or need too much precision for traditional CRISPR-Cas9 with its reliance on HDR. Prime editing can address these types of genetic errors with, frankly, unprecedented accuracy. It offers a path to correct the exact defect without the larger cuts, or the limitations of relying on the cell's sometimes inefficient HDR pathway. And this means, hopefully, a higher likelihood of successful therapeutic outcomes with minimal unintended changes, which is obviously paramount when you're dealing with beloved animals, with pets.
Okay, so we've unpacked the molecular machinery, and it's incredible how precise these tools are becoming. We're moving from, broad strokes to incredibly fine, detailed work. But the real magic, I suppose, and also the real challenge, lies in applying these microscopic corrections to living, breathing animals, right? So let's try and bring this science to life with some concrete examples from the research, illustrating what this all actually means for our pets. Because we're not talking abstract concepts here. This science is actively being applied to very real animal health problems. Let's take, for instance, progressive retinal atrophy, PRA, in dogs. This is a truly devastating inherited condition. It leads to progressive degeneration of the retina and ultimately blindness. We see it in many popular dog breeds, Labradors, Golden Retrievers, Poodles, many others. The dogs, they just gradually lose their vision, often becoming completely blind within just a few years. It's absolutely heart-wrenching for the animal and, of course, their owners. Now, its genetic basis, it's often linked to specific point mutations in certain genes, like the PDE6B gene, for example. That gene plays a crucial role in the function of photoreceptor cells in the retina. So, in principle, with a technology like base editing, you could directly target and correct that single specific mutation, that one typo, maybe a specific G changed to an A, that's causing the disease. I mean, imagine the impact. Restoring or just preserving sight for these animals.
And the application level here is, as you hinted, profoundly important, not just for treatment, but for the future of animal breeding and welfare, too. This kind of genetic correction, it could potentially be applied at either the somatic level or the germline level.
Okay, somatic versus germline. What's the key difference there?
Right. So if you apply it at the somatic level, you're treating the individual animal, the one already afflicted with or maybe predisposed to the disease. You're aiming to stop or slow the disease progression within that one dog. This would involve, say, delivering the gene editing machinery to the retinal cells to correct the mutation in the affected tissue. This offers immediate therapeutic benefit, hopefully, to that individual animal. But if you apply it at the germline level, that means editing the embryo or maybe the reproductive cells themselves, the sperm or egg cells, then the genetic correction becomes heritable.
Heritable, meaning it gets passed down.
Exactly. It would be passed down to all future generations, potentially eliminating the disease from that dog's entire lineage forever. So for a breed plagued by a specific inherited blindness like PRA, this could mean that future generations are just born entirely free of the disease, which represents an incredible long-term welfare gain. Of course, the ethical implications, as we'll definitely discuss later, are really significant here, precisely because of that permanence.
Exactly. That permanence factor is huge. And another powerful example comes from the feline world, hypertrophic cardiomyopathy, HCM, particularly in Maine Coon cats. Now, this is actually the most common heart disease in cats overall, but it's particularly prevalent and often quite severe in Maine Coons. It's a progressive disease. The muscular walls of the heart's main pumping chamber, the left ventricle, they become abnormally thickened. This reduces the heart's efficiency over time, and it can lead to heart failure, dangerous blood clots, and tragically, sudden death. And its genetic basis, well, it's been clearly linked to a specific mutation in a gene called MYBPC3.
That's right, the MYBPC3 gene. It codes for a protein that's essential for normal heart muscle contraction. And affected cats, they often show symptoms much later in life, which makes it kind of a hidden threat in many breeding lines. So the profound implication here, building on the germline idea, is the possibility of correcting this single defective allele, this specific MYBPC3 mutation, right at the embryo stage. Imagine intervening when the animal is just, a tiny ball of cells, perhaps even before it's implanted in the mother. By performing germline editing at this earliest stage, you could potentially prevent the disease from ever developing in that animal and critically prevent it from ever being passed down to its offspring.
So you could, theoretically, wipe it out of a line completely.
Theoretically, yes. If successful and applied carefully across breeding programs, this could literally eliminate the disease from entire bloodlines, and eventually maybe even from the breed as a whole. This really demonstrates the powerful heritable impact of germline editing. It's not just about treating one animal, it's about fundamentally altering the genetic predisposition of a breed for generations to come. It's about preventing future suffering rather than just managing a lifelong debilitating illness. I mean, the thought of a future without HCM and Maine Coons, that's incredibly compelling.
Yeah, and when we talk about the future, the promise of gene editing in companion animals, I think the most profound and immediate benefit that really jumps out is exactly that, the potential to eradicate inherited suffering. We are talking about moving beyond just managing the symptoms of these chronic, painful, or debilitating conditions, moving towards actually eliminating the diseases right at their genetic source. This isn't just about making pets, a bit more comfortable.
It's potentially about preventing untold generations of suffering for these animals and also the heartbreak for the families who love them. Imagine, for instance, a future where polycystic kidney disease, PKD, a significant and often fatal issue in Persian cats and related breeds too. Imagine it could be forever eliminated from the breed through targeted germline editing.
PKD, that's the one with the cysts in the kidneys.
Exactly. PKD is characterized by the formation of multiple fluid-filled cysts in the kidneys, and they progressively enlarge, they damage the kidney tissue, and ultimately they lead to kidney failure. It's common, it's painful, and often it's a silent disease until it's quite advanced, and it definitely shortens lives. Think about the countless cats and their families who currently suffer from this, requiring lifelong management, special diets, medication, and ultimately facing a pretty grim prognosis. Gene editing offers a path, potentially, to ensure that every kitten born into a line previously affected by PKD could be genetically free of it from day one. This represents a massive, truly enduring welfare benefit, preventing suffering for countless future generations of animals. It's a fundamental shift, really, from reactive treatment to proactive prevention right on a genomic scale, changing the very genetic landscape of these beloved breeds.
And what's really crucial to understand here, I think, is that this isn't just theoretical. This isn't merely a maybe one-day scenario confined to a whiteboard or some distant dream. There's already published, peer-reviewed progress out there that vividly demonstrates the real-world potential. We're seeing tangible results being achieved. The science is moving incredibly fast from hypothesis towards demonstrable proof of concept. For instance, you mentioned Duchenne muscular dystrophy, DMD, earlier. Canine models of DMD have actually shown measurable improvement in muscle strength and overall health after CRISPR treatment.
That's right. DMD is a severe progressive muscle wasting disease. And in dogs, particularly golden retrievers actually, it mirrors the human condition very closely. It causes severe weakness, difficulty walking, and often sadly leads to early death due to respiratory or cardiac failure. The research has successfully demonstrated that by delivering CRISPR components to muscle cells, the underlying genetic defect can be, well, at least partially corrected, leading to more functional muscle tissue.
So it's working, at least to some extent.
Yes, it's not a complete cure in these initial studies, let's be clear, but it's a significant proof of concept in these animal models. It shows that the technology can indeed have a measurable positive impact on a systemic disease like DMD. Now, those dogs are still research animals, of course. No veterinary clinic is offering widespread CRISPR therapy to pets right now. But this kind of progress, it clearly illustrates the translational research stage we're in. The trajectory is clear. Establish proof of concept in animals, then refine delivery systems, improve efficiency, ensure safety, and eventually move towards clinical applications for our companion animals. Those very tangible gains in muscle strength observed in these dogs, they're really a beacon of what might be possible. And building on that, gene editing offers this potential to truly revolutionize animal welfare, to move us beyond that traditional approach of just managing symptoms, which is often all we can do now. For far too long, we've sort of accepted certain breed-specific health problems as just part of the breed, an unfortunate trade-off perhaps for desirable aesthetic treats or certain behavioral characteristics. But gene editing fundamentally challenges that notion. It offers a way, potentially, to retain the beloved characteristics while eliminating the suffering that so often comes packaged with them. Take the widely recognized example of bulldogs. English bulldogs, French bulldogs. They often suffer from severe breathing problems due to the brachycephalic syndrome.
Right, they're flat faces.
Exactly. Their distinctive flat faces, the shortened snouts, the altered skull structure. These anatomical features, while, yes, endearing to many people, they result in narrow nostrils, elongated soft palates, constricted airways, leading to chronic respiratory distress, problems with overheating, and just significant discomfort for the dog. Traditionally, the solution involves invasive surgeries, widening nostrils, shortening soft palates, removing excess tissue in the throat.
Which are serious surgeries.
Absolutely. Painful procedures aimed at alleviating symptoms, improving quality of life. But they don't address the root cause, the underlying genetics. However, instead of performing surgery after surgery throughout a bulldog's life, gene editing offers the profound potential to edit out the underlying genetic predispositions that cause their breathing problems in the first place. Perhaps by subtly altering the genes that control facial bone development right at the embryonic stage. This could mean preventing those anatomical defects that lead to respiratory distress from ever even occurring.
So, bulldogs that can actually breathe properly from birth.
That's the hope. This isn't just a minor improvement. It could be a true revolution in animal welfare, preventing the problem from ever occurring rather than just treating its manifestations throughout an animal's life. Imagine bulldogs that can breathe freely, play without gasping for air, live long, comfortable lives without needing multiple corrective surgeries. That's the kind of transformative power we're talking about here.
Okay, but every powerful technology, no matter how revolutionary it seems, it comes with complexities, right, and risks. And gene editing is certainly no exception. While the precision of these tools has, as you said, dramatically improved, there are still significant scientific and practical hurdles to overcome before this becomes a widespread, safe, and truly effective reality for our pets. It's like having a very powerful scalpel. And with that power comes immense responsibility and numerous challenges to perfect its use. One of the primary and I think most critical concerns is this challenge of off-target effects. Could you explain that a bit more? What are off-target effects?
Absolutely. Off-target effects refer to the possibility that the gene editing machinery, despite being designed for a specific spot, might actually make unintended edits elsewhere, meaning it cuts or modifies the wrong place in the genome, a place you didn't intend to change.
So hitting the wrong target.
Exactly. And even with significant improvements in the Cas9 variants, people have engineered versions like ESP-Cas9 or high-fidelity Cas9-HF1 specifically to reduce these errors. The risk still remains. It hasn't been eliminated entirely. Now, in a companion animal, such off-target effects could potentially introduce entirely new, unpredictable diseases or health issues. These could, theoretically, be worse, more complex, or harder to treat than the original condition you were trying to fix. You could turn a hopeful intervention into an unforeseen and perhaps tragic disaster. Researchers are constantly refining guide RNA design, improving the Cas9 variants, developing better detection methods. But ensuring absolute specificity across a vast and unique genome like a dog's or cat's, it's incredibly complex. Which is why the pursuit of ever higher precision and robust screening methods to detect these unintended edits are so critical in this field. We have to be sure we're not inadvertently trading one problem for another, potentially worse one. And then, beyond the precision of the edit itself, there's a major practical challenge, especially for systemic diseases, things affecting the whole body. It's what we often call the delivery bottleneck.
The delivery bottleneck, okay.
Yeah. This refers to the immense difficulty in actually getting the gene editing tools, the CRISPR components, the guide RNAs, maybe repair templates if you're using HDR, getting them to the right place in the animal's body at the right time, and crucially, into enough cells to actually make a meaningful therapeutic difference. Imagine having the perfect key, the perfect editing tool, but you have no way to get it to the lock. Or maybe you can only get it to a few of the millions of locks that need opening. It won't be very effective. Let's illustrate this complexity again with Duchenne muscular dystrophy, DMD. As we said, the genetic mutation affects skeletal muscle fibers throughout the entire body. These are countless cells spread across large muscle groups everywhere. Now, while adeno-associated viral vectors, AAVs, have shown some partial success in delivering CRISPR to canine models, they come with their own set of challenges.
AAVs, those are like modified viruses used for delivery.
Exactly. They're like tiny natural delivery trucks. They're often used because they typically don't cause a big immune reaction in the recipient, which is good. And they can slip into many different cell types, including non-dividing ones like muscle cells. However, they have a limited cargo capacity. They can only carry so much genetic material, so fitting the entire CRISPR system in can be tricky. More importantly, Getting those vectors to effectively reach and modify every muscle fiber in a large animal, or even just a clinically significant percentage, enough to have a widespread therapeutic effect. That is a truly daunting task. The sheer volume of viral particles required for systemic delivery in, say, a large dog, ensuring their uniform distribution throughout such a vast and complex system, and navigating the animal's natural immune responses to the viral vector itself, it's immensely difficult.
So just getting the medicine to where it needs to go is a huge hurdle.
A massive hurdle. It's akin to trying to precisely paint millions of individual tiny cells across a vast canvas, ensuring each one gets the exact shade you want, without missing a single spot, and without the paint itself causing an allergic reaction. The logistical and biological hurdles are immense.
Okay, so delivery is tough. But even if the delivery system is perfected, right, you get the tools to the right place. You mentioned another challenge that researchers frequently encounter, particularly with embryo editing experiments, mosaicism.
Yes, mosaicism, that's another significant hurdle.
What exactly is mosaicism in this context?
Mosaicism means that not every single cell in the target tissue, or sometimes even the entire organism, successfully incorporates the intended genetic edit. You might end up with an animal where some cells have the corrected gene, while other cells still carry the original, unedited, faulty gene. It's a patchwork.
Ah, so it's inconsistent editing across the body.
Precisely. This phenomenon arises because the editing might not occur in all cells simultaneously. Or maybe some cells successfully repair the break using the desired pathway, while others use a different pathway or fail to repair it correctly. Especially if the editing happens a bit later in embryonic development after the first few cell divisions have already occurred. And this phenomenon, it's been widely observed in embryo editing experiments across multiple species, from mice all the way up to larger mammals. Now, for gene editing to be truly effective in a clinical setting, especially for inherited diseases where you really need a high percentage of cells corrected, you need high levels of editing efficiency across the affected tissues. Mosaicism really risks leading to incomplete cures, meaning the animal might still experience some degree of suffering from the original disease despite the intervention. This reduces the overall efficacy and frankly raises questions about the true benefit to the animal. It could potentially create a partially cured state that's still far from ideal. And leading on from that, there's perhaps a more fundamental, maybe even humbling concern, which is the inherent interconnectedness of biology itself. We must always remember that biological systems are incredibly complex, intricately interconnected networks. So when we intervene at such a fundamental level, editing a specific gene, we absolutely have to ask, what if editing out one genetic problem inadvertently introduces other weaknesses or vulnerabilities elsewhere in the organism?
You mean like unintended side effects from the change itself, not just from errors in the process?
Exactly. This is often referred to as pleiotropy, where one gene influences multiple seemingly unrelated traits. So this could manifest as, say, unexpected developmental defects that weren't present before, or perhaps immune system dysfunction, or even subtle yet significant metabolic issues that only become apparent over time, maybe under certain stressors. For example, some genes that are linked to a specific disease might also play crucial roles in other biological processes that we don't fully understand yet, or they might be part of complex regulatory networks. Altering one part of this intricate web, even with the best intentions and the most precise tools, it could potentially have cascading, unpredictable effects throughout the animal's biology.
Because biology isn't simple, one gene doesn't just do one thing.
Precisely. The core principle to consider is that evolution is a highly integrated process. Millions of years of natural selection optimizing for survival in complex environments. One gene rarely acts in isolation. Genes interact in complex pathways, influencing everything from cellular function to organ development, behavior, even susceptibility to other diseases. Our understanding of these vast, interwoven biological networks, while it's rapidly expanding, is still far from complete, very far. So to intervene in such a fundamental system requires profound humility and a rigorous, long-term approach to monitoring any potential downstream effects.
Okay, these technical hurdles are significant, and they lead us directly, I think, to the even more profound ethical questions. Questions that gene editing in companion animals inevitably raises. Because once these incredibly powerful tools for rewriting the genetic code exist, and as their precision improves, how do you prevent the creation of what some people are already calling designer pets? This isn't just a theoretical debate anymore, is it? It feels like a very real concern that quickly moves beyond pure medical necessity. I mean, there will inevitably be significant pressure, won't there? From certain breeders, maybe even from new biotech startups, pressure to move beyond purely therapeutic applications into cosmetic edits or edits for other desirable but non-medical traits.
That pressure is almost certainly going to exist, yes.
So we're not just talking about preventing disease anymore. We're potentially talking about altering traits simply for human preference, creating animals that are tailored to specific human desires rather than focusing solely on their intrinsic well-being. Examples could include, I don't know, engineering non-shedding coats for certain breeds because people don't like vacuuming, or creating truly hypoallergenic cats for allergy sufferers, which, okay, might have a health link for the owner, but not the cat. Or even purely aesthetic modifications driven entirely by market demand or human whims, changing ear shape or size or coat patterns in ways that serve absolutely no purpose for the animal's health.
These are all plausible scenarios down the line.
So the dilemma here feels stark. Does the pursuit of undeniably life-saving applications like curing PRA or HCM, does that justify opening the door to the potential commodification of animals, where their genetic makeup is manipulated just to fit human desires, rather than their inherent health needs or their natural blueprint? Where do we possibly draw the line between compassionate intervention and, well, genetic customization?
And this leads us directly to that crucial distinction we touched on earlier, germline editing. This is really where the ethical stakes become the highest and where the discussion gets most intense and, frankly, most difficult. Because, as we said, the effects of germline edits are not confined to the individual animal being treated. They are heritable, meaning they're quite literally passed down through all future generations of an animal's lineage. It affects the offspring, their offspring, and every single subsequent generation after that.
That heritability factor just changes everything.
It really does. It introduces a profound level of permanence and potential irreversibility. If an unintended negative consequence, an unforeseen problem like a subtle predisposition to a new disease, or an unexpected developmental issue if that spreads through a breed via germline editing, reversing it becomes nearly impossible. You can't just recall it. You haven't just affected one animal. You've potentially altered the genetic destiny of an entire population, possibly for centuries to come.
Wow. Altering the course of evolution, essentially.
We would essentially be altering the course of evolution by direct human choice, with potentially unforeseen long-term consequences for entire animal populations, maybe even for their interactions within ecosystems or their long-term resilience to environmental changes. This is a level of intervention that demands extreme caution, rigorous long-term studies before any widespread application, and a profound sense of foresight. The implications, they're not just for the individual pet sitting on your lap, but for entire species and their future genetic health.
Now, some people might draw parallels here. They might look at other groundbreaking but initially controversial technologies, like in vitro fertilization, IVF, for example, which faced significant outcry when it was first introduced for humans, but now it's considered pretty standard practice in medicine. They might argue, well, society will eventually accept gene editing for pets in the same way, just another advanced reproductive technology.
While it's true that IVF also faced intense ethical debates, and that comparison is often made, I think it's really critical to nuance that comparison. It's not quite apples to apples. IVF, at its fundamental core, it facilitates reproduction, it assists the fertilization process, it helps overcome infertility, or allows for screening of embryos for existing genetic conditions, but it doesn't rewrite genomes. It doesn't alter the genetic code itself.
Right. It works with the existing genetic material.
Exactly. CRISPR and gene editing technologies in general fundamentally do rewrite genomes. They make intentional changes to the DNA sequence. This is not just a technological step, it's a huge qualitative leap in the level of intervention. And this fundamental difference means that mistakes, especially with germline changes, are permanent, irreversible, and can be far more widespread and impactful than any issue likely to arise from IVF. This makes the risks far greater, in my view, and demands a different, arguably far higher level of ethical scrutiny. We are not just assisting reproduction here, we are potentially redesigning the blueprint of life itself, and that carries a much, much heavier burden of responsibility. Which brings us, I think, to the central philosophical conflict in this whole field. What I see is the core tension between, on the one hand, compassion, and on the other, humility.
Compassion versus humility. Explain that.
Well, on one side, we have this powerful, almost irresistible drive of compassion, our empathy. This is the desire to intervene, to alleviate preventable suffering. like the really compelling example of bulldogs struggling to breathe or cats suffering from painful kidney disease. The ability to potentially eliminate such suffering right at its genetic root is an incredibly powerful motivator. It promises a future of healthier, happier lives for our animal companions, a future where they could be free from the burdens of these horrible inherited diseases. And this drive, it comes from our deep bond with our pets, our desire to do what's best for them. But then, on the other side, we have the profound need for humility. Humility before complexity. This is the urgent call for extreme caution, for acknowledging the vast unknowns of these complex biological systems. It's understanding that our current knowledge, while it's expanding rapidly, is still far, far from complete. When we alter a gene, yes, we may fix one problem, but what about those unintended consequences we talked about? Could that gene play some unknown role in resilience to other diseases? Or in subtle aspects of behavior or development that we just don't appreciate yet? There is always the potential for unforeseen harm when intervening at such a fundamental level, when we're dealing with millions of years of evolutionary fine-tuning.
So it's the desire to help versus the fear of messing things up inadvertently.
Exactly. To refuse the technology outright because it might be misused or because of unknown risks, well, that means letting preventable suffering continue, which goes against our compassionate instincts. But to proceed without rigorous safeguards, without deep critical thinking, rushing into poorly understood interventions, that's equally dangerous, risking new and potentially irreversible forms of suffering. Both perspectives are valid, they're deeply felt, and they are absolutely essential to this discussion. Finding the right balance between these two powerful forces, compassion guiding us to act, and humility urging caution, that's the ethical tightrope we as a society must collectively walk. #5. Regulation and societal decisions.
Okay, so if the science itself is this complex, this intricate, and raises such challenging ethical questions, then the natural next question is, what about the rules? What about the regulations governing it? How is this incredibly powerful technology being managed and controlled, particularly when we're talking about applying it to companion animals? Well, unfortunately, the current regulatory picture for gene editing in companion animals is, well, frankly, it's a bit of a mess. That's the consensus. It's highly fragmented. It's incredibly inconsistent across different jurisdictions, different countries, even different organizations within the same country.
Yeah, there's a real lack of consensus.
Breeding clubs, national and international veterinary boards, government agencies, they simply do not yet agree on what is permissible, if anything, when it comes to gene editing pets. And this contrasts really sharply with the situation for, say, livestock, food-producing animals.
That's a key difference.
For those animals, there are established frameworks from bodies like the FDA in the US, the European Medicines Agency, the EMA over here, primarily because there are clear concerns about food safety, about human consumption of those animal products.
Right, there's a direct human health and commerce link.
Exactly. And that clear regulatory impetus is largely absent for pets, which leaves a significant void in oversight. This fragmentation means a therapy or a modification that might be deemed unacceptable in one region could potentially be pursued in another, creating this kind of patchwork of standards that's difficult to enforce or even just to track. And you add in the emotional attachment we have to our pets, combined with the diverse values and motivations of pet owners, breeders, the scientific community, it just makes this regulatory landscape particularly challenging to try and unify.
And this situation, it brings us to an inherent danger, one we often see with these kinds of transformative technologies. It's when scientific progress significantly outstrips the development of the ethical and legal frameworks needed to manage it.
The science moves faster than the rules.
Much faster. The speed of scientific advancement in gene editing is truly breathtaking. The technology is rapidly outpacing the creation of necessary safeguards and oversight. New techniques, new applications, they emerge faster than societies can properly deliberate, debate, and legislate. And this creates a really precarious situation, where powerful capabilities exist before society has fully grappled with their implications, leading to potential Wild West scenarios or unforeseen consequences that we didn't anticipate. Now, we must reiterate, rigorous, transparent, long-term research is absolutely essential. No responsible scientist would advocate for rushed, poorly characterized edits being used clinically right now. The calls for responsibility from within the scientific community itself are strong. Many researchers are advocating for caution, for careful, stepwise progression. However, the regulatory bodies and the broader societal discussions, they really need to catch up to ensure that this powerful tool is used wisely, ethically, rather than recklessly or for purely commercial, non-welfare-driven purposes. Without robust oversight, the potential for misuse and unintended harm, it just increases exponentially.
And that's why this isn't just a discussion for scientists in a lab somewhere or for regulators behind closed doors. It's profoundly a societal conversation. It demands broad public engagement. It extends far beyond the confines of academia or biotech companies. It's absolutely vital that we stress the importance of involving diverse voices in shaping these decisions. That includes scientists, yes, who understand the technical capabilities and limitations. It includes veterinarians who see these animals daily, understand their welfare needs, the practical realities of pet health. It includes ethicists who can help us navigate the moral complexities, the long-term implications. And crucially, it must include pet owners themselves, people like you listening. Pet owners were on the front lines of animal companionship. They will directly experience the consequences, both positive and negative, of these technologies.
Their perspective is invaluable.
Absolutely.
Yeah.
The reality is, gene editing in companion animals, it's no longer some niche lab issue confined to academic papers. It's rapidly becoming a societal concern, one that demands broad public engagement and informed dialogue, because the implications, they extend far beyond the laboratory. They touch on our deepest values about animal welfare, about companionship, even about our role in directing evolution. We need to reinforce that the first gene-edited pet therapies, they could arrive within the next decade, maybe sooner for some applications.
It's not science fiction anymore.
No. Which means the decisions we collectively make now, or perhaps more dangerously, the decisions we fail to make will have significant and lasting consequences for animal welfare and for our fundamental relationship with our pets. The time for these conversations is absolutely now.
Indeed. And for you, the listener, thinking about all this, gene editing in companion animals isn't some distant abstract concept anymore. It's not just a future possibility. It's becoming an imminent reality. It's coming. And society, all of us, truly has to decide where the line is, or where the lines are, perhaps. The decisions we make now about its applications, about its regulations, they matter immensely for the future of animal welfare and for our fundamental relationship with our pets.
Thank you so much for joining us on this deep dive into gene editing in companion animals. It's clearly a field brimming with both immense scientific promise and also profound, complex ethical questions that will undoubtedly shape our future.
And as you reflect on everything we've discussed, maybe consider this final thought. If we gain this ability, this power, to precisely direct evolution, to alleviate suffering right at its genetic root, what new responsibilities does this unprecedented power place upon us as humans? And how might it fundamentally redefine our relationship with the animals we call our companions moving forward? Keep questioning, keep learning, and please keep thinking critically about where science is leading us.